Well, this is our second set of lectures that we're calling Allelic Interaction. So this is Allelic Interactions 2. This is the first part of that. We'll have two more parts, so part 2 and 3. Um, and really, I'm going to call it, in our case, more allelic interactions. So we're really talking about additional modes of inheritance that relate to the fact that when you get two alleles, particularly in the heterozygous condition, there has to be some interaction between those in some way. Okay. Now, this series of lectures, and actually those that follow this, are really the ones that tend to be complicated for students. So really need to pay attention to what I'm saying here, and you really need to go look and read in the book and think about what's being said here, because it's going to kind of torque your thinking just a little bit. Plus, we're going to throw a number of terms at you from now on. But underneath that, remember, it's simply Mendelian, right? The crosses we make are the crosses. They're the same. They're not going to change from Mendel's Big Six. What's changing is the way that phenotype is derived from those and the effects that these alleles have both on each other as well as ultimately on the phenotype. All right, so let's start with our first kind of new term right here, okay? And we're going to talk a little bit about things might be coat color as we see them in animals and plants. This is a really nice little mouse opossum, species of mammals, small mammals found Mexico and into Central and South America. And little prehensile tail, as it's called, they can wrap around. They're really cute little guys. So there's the possums. The reason I have them up here is that coat color in mammals, the color of the pelage, is often affected by multiple genes, but it's also sometimes, in many cases we're going to illustrate here, one gene can affect lots of parts, okay? So we've kind of got multiple things going on. Right now we're going to concentrate on this idea that we've got a single gene affecting multiple parts. We're going to call that activity pleiotrophy. Sometimes it's called pleiotrophy, depending on the part of the world you're from. But pleiotrophy is the idea that you can have multiple phenotypic effects from a single gene. Basically, you've got some gene and it has some outcome. And this makes perfect sense if we go back to thinking about previous lectures when we said really what alleles do, what genes do, is contribute to biochemical pathways, right? Well, biochemical pathways tend to be things where they branch and branch so that you get multiple effects. Well, obviously things like a good example here is color, because the melanin and our other pigments that are involved in color are often part of fairly complex pathways. So you're not just taking a gene and making, color on the head, or making a gene and making color here. That making that color may in fact impact eye color, hair color, all sorts of things. In our little simple example here, we've got a case where we've got our little beach mouse, right? And there are some known genes here. There are several genes that control color, but there's at least one gene that affects two parts of the color. The first one, this one gene, this one locus affects the head pattern. And this one, you can see, has a fairly reduced head pattern here. So that we're talking about this little dark area between the ears and right down between the eyes. This is with the same species, but a different subspecies here, and has a different genetic combination. And so you see that the colors go all the way down into the nose part. Okay, so the head pattern here. Now, they also have what's known as a tail stripe. Right on the top of the tail here is a little stripe that goes down the tail. Okay? And that can vary quite a bit. The head pattern is pretty much a dominant recessive system. You either have this recessive form like this, or you have this dominant form where you have mostly large expression. And there's some more complicated genetics here, but for our purposes, this will do, okay? So what we're talking about here is we're having a single gene and it's controlling color, right? But the placement of that color is relative to the head. Here it's a dominant recessive. At the same time, that gene is producing pigments that are on the tail, and on the tail we have incomplete dominance because we have the almost complete lack of a tail stripe as seen in this guy. And then you have tail stripes which can be like a quarter length, 3/4 length, or a full length. This was a full length one here, but they can be a little shorter, and it may be within the same population, but you can have different lengths. So you kind of have short, you have kind of no tail stripe, a short tail stripe, or a full tail stripe. So essentially it's incomplete dominance in some form, okay? This is pleiotrophy, just a silly example here where we're talking about the fact that you got a single gene controlling lots of things. And you can think about a gene that might control something like cartilage. right? Production of cartilage. Well, that's going to affect your nose because that's got cartilage. It's going to affect your ears, which has cartilage. It's going to affect lots of other areas where cartilage is found. So it's one gene having an effect on multiple phenotypes. All right. Now we're going to complicate this a little bit because we will need to talk about a certain types of modes of inheritance that involves very specific traits. And what we're going to talk about here is the idea of pleiotrophy, where it affects a normal trait, but then the same gene can have effect on whether you live or not live. Okay, so we're going to say that one of the expressions is, in our case, life. So this is an extreme case, right, where a gene is sort of affecting a normal trait, and then it also controls whether you even live or not. Well, we would, of course, call these lethal genes. That's what they are, right? That's the idea we're working under. A lethal gene is any allele that causes death, right? Results in death. Or from a biological perspective, and particularly an ecological perspective, it means it prevents reproduction. If you're not carried into the next generation, then essentially you're lethal if you can't move forward. So technically, lethal genes were considered those that would kill you. But if you can't reproduce, then in the terms of an ecological sense, it's the same as being a lethal. All right. Now, very, very important. Pay attention to this part right here. Okay. We need to really think about the true meaning of dominant recessiveness. Okay. True meaning of what it means to be dominant recessive. Okay. What do I mean by that? All right. A recessive allele, what does it mean to be recessive? It means it takes two copies to be expressed. That's all it means. Okay. Doesn't matter if you call it big A's or little a's or pluses or whatever. It takes two copies in order for expression to occur. Right. A dominant allele is one where you only need one copy. It doesn't matter how you express it as a big S or a little q or whatever. However you express it, it only takes one copy for that to be expressed, right? We would say haplosufficiency if we're going back several lectures about a term that we might use. Okay. Now, why do I say that's important? Because we're going to talk about certain types of lethals that relate to dominant and recessive, and people just start to confuse these a little bit because of they're so used to these little letters being recessive and the big letters being dominant, and they just mentally have had that for so long that it gets intertwined with what we're talking about. And it's one of the reasons that I've been trying to be very careful and not always use these big letters and small letters. Remember when we did codominance and we talked about things like roan, I tried to use red and white allele, a big R and a big W, so that you would think about it in terms of that. And instead of having big A, little a, which works fine. I mean, it's fine to note them that way, but again, we need to think about what it means. What it means to be recessive is you got to have two copies. What it means to be dominant is you only need one copy, okay? Now, where we're going to play a role in that is when we talk about lethals and the types of lethals that you have. A recessive lethal is any lethal allele that only expressed when it's in the homozygous condition, okay? So think about it in terms of pleiotrophy. It's an allele that may be off over here controlling color in some codominant fashion or incomplete or dominant, whatever. If you get two copies of that allele, it kills you. Okay, that's what it means. Doesn't matter what it's doing over here as far as color, it means that you need two copies of it to kill you. Conversely, a dominant lethal is this. It's one where you only need one copy to kill you. Therefore, it's expressed both in the homozygote and the heterozygote. Now, once again, it doesn't matter what it's doing, right? The pleiotrophy part, it could be off over here controlling, anything, right? Hair color or eye color or whatever, leaf shape. But if you get a copy of that allele, that mutation, it's going to kill you. That's called a dominant lethal. So what I'm doing in this lecture is talk about dominant lethals, and then we will talk about recessive lethals in the next part of this lecture, part 2, and then we'll work some problems and do some other things, okay? So let's concentrate on dominant lethal. We've got 3 genotypes here, right? We're going to make the little a's, and for this case, a lethal, okay? And we're going to make that little a dominant lethal, which means you only need one copy to kill you, which means literally both of these genotypes are lethals. Okay, here you're just kind of double dead, right? I mean, you're just basically dead if you have this. And in this particular case over here, you're heterozygote, but even if big A is making you orange or something, right? It's controlling your color. If you get a little a, which might be making you white, it not only top of making you white, it kills you, right? So this is a dominant lethal. Now, obviously, if you mutate to a dominant lethal, it's only going to last one generation if it's a true dominant lethal, using some air quotes there. Because if you mutate to it and maybe in gametic formation, you pass it on to your kids, they die, right? If they get even one copy. So true dominant lethals are only around one generation, okay? But there are lots of dominant lethals, okay? So the big question is, how in the world can we have dominant lethals if they only last one generation? Well, obviously they last more than one generation. And how can they do that? Well, there's a number of ways, and I'm going to kind of make them in very simplistic, kind of strange terms here that you might not see in a book, but I think will explain what it would mean. Okay. The first idea is I'm just saying genes cheat. Okay. And how do I mean cheat? How can a gene cheat at anything? Well, there's several ways I mean by this. I mean, there's even theories about it. There's theories of what's called the selfish gene theories. And the selfish gene theory basically says, and it goes back to the gene's job, individual job, is to get into the next generation, right? And it will cooperate with other genes to do that. It'll cooperate with organisms or whatever, but its job is to get to the next generation. Okay, if it kills you immediately, it can't get to the next generation. So somehow it has to figure out a way to cheat, right, and get around that limitation. So what are some examples of cheating in the terms that I'm trying to use them here? One obvious one is one called delayed expression. Delayed expression is very simple. It's when, yeah, it's going to kill you, okay, if you're homozygote or heterozygote, but it waits long enough for you to reproduce. That's basically the idea, okay? A good example of that might be Huntington's disease, Huntington's chorea. It's a debilitating neurological disease. And this is sort of an example of what happens to the brain. This is a normal brain here, okay? And this is a person up here with Huntington's disease. It basically just destroys enough of the neural system that eventually you die from it, right? And so it becomes a dominant lethal. Uh, it's fairly rare, of course, but individuals that are, most of us are normal, okay? If you get one copy of that allele, we'll call it little h, the allele, you will develop the condition, but you generally won't develop it until you're into your 40s. Now, for most of us, if we're going to reproduce, we've probably reproduced by the time we're 40. You know, we may continue, but we've certainly, that will be the time that we have reproduced. So it waits long enough in the heterozygous condition that it can persist. Now, in the complete recessive condition, it's usually an early lethal, so you may not even make it through fetal development or whatever. Okay, it's a true lethal there, and you know, if you make it even to live birth, it's not a long-term system. But the individuals that we see in real human populations are these individuals that have a normal allele and then one of these dominant lethal alleles, but they are carriers. They can pass them on to their children, okay? And you can think about most of the crosses that you would make. This is a rare disease, so most of the individuals that have it are heterozygotes that are living, because these don't live usually to adulthood. They're usually mating not with another het, okay, but usually with a normal individual. So about half the progeny are going to be completely normal, and about half are going to carry this developmental condition for Huntington's disease, right? Um, Famous Woody Guthrie, famous musician that wrote, you know, "This land is your land, this land is my land," and things, and many other folk songs. Was a very strong activist in the 1930s and 40s and into the 50s. Died of Huntington's chorea. His son Arlo Guthrie, who is still a musician, a famous musician, you know, is worried. Do I have it? So of course they can have their stuff tested. So there are testing systems to see whether or not you have it. Arlo's well past his 40s and did not have was not a carrier for it, but it get tested. Okay. Um, Now, another part that plays into cheating, I guess, here is what would be called genes that are sublethal or semi-lethal. They're terms that you will see. Um, Sublethal means that it's not fully lethal, okay? It may debilitate you or whatever, and it may eventually kill you. And sometimes Huntington's considered that way because you know, it's a slow debilitating system. Semi-lethal means that not everybody that gets the allele dies from it. And we'll talk more about that in a moment in terms of penetrance. But semi-lethal It's just, the whole population that may have the allele, even though it's going to kill you, it has the capability, doesn't quite do that. All right, and we'll talk a little bit about that in a moment. But let's think about those terms in the other way that you can have this system. So if we take delayed expression is one way that they can cheat. The other way that they can cheat is what's known as a conditional lethal. All right, conditional. Now notice this word conditional. I'm going to use it a lot. Okay, it has a very simple meaning for us. It means that there's some environmental influence on the expression of that particular allele or that particular mutation. Okay, in our case, there's an environmental input on effect on whether it's lethal or not. Okay, that's the extreme case. Almost all alleles are somehow have a conditional part to them. Every expression to phenotype, the way we look, has both a genetic component and an environmental component. Now, it could be the external environment, right, like temperature of the room or water or what you have as far as nutrition, or it can be intercellular environment. Either one of those are considered environments. So those conditions affect what's expressed. A conditional lethal is a good example of this, okay? In a conditional lethal, you only get expression when you get certain conditions that apply. One example might be something like fruit flies that become involved with there's a HDA, there's an alcohol dehydrogenase system in fruit flies that affects whether or not they live or die, basically. There's a temperature dependent mutation systems in fruit flies. If you raise fruit flies at a certain temperature, they do fine. If you lower that temperature maybe to 15 degrees C or so, then you'll have lethality, okay? But the fruit flies, they need a lot, they use fruit where they lay their eggs, and this is an example here of a cherry. And the cherries often water will come down in the stem and sit here and ferment because there's natural sugars, right? So you basically sort of making a cherry wine on these systems. And fruit flies learn to avoid certain amounts. They can tolerate some amounts and they can't tolerate others, And these fruits though, and bananas can do the same thing. I mean, fruit flies, they lay their eggs in fruit, so they use all sorts of things. But these cherries, good example. That particularly if you're anybody for spending time up in say Montana, northern Montana, and there are lots of cherry farms. There are often pie cherries up in that region of Montana and Oregon and Washington. A lot of sugar. The pickers come through, they pick, but not all of them are ready at that time, so they leave some on the trees. It starts to get wet, water drips down into there, and you end up with cherries with lots of actual alcohol. In fact, it's a big problem because bears will come in and down and they'll feed on these cherries and they'll get drunk and they'll break the limbs out of the trees. And it's not just that. I mean, the famous Marlu trees that are found in Africa, this big, beautiful fruit. The fruit ferments. Peoples of Africa make alcoholic beverages out of them, but the animals will come along and eat them too, and they'll end up being, you see drunk elephants staggering around and busting through gardens, and monkeys will get drunk on it and stagger around. It's a quite common sight when these fruits are actually there. Now that's not lethal. I mean, it's not going to be lethal to the elephant unless it does something bad, or the bear unless it gets in and gets shot. But they're conditional, right? The system. So why am I talking about all this? Because if we go to fruit flies and we think about this alcohol issue, this naturally occurring alcohol, there are mutations in fruit flies in the alcohol dehydrogenase system where they either can or can't effectively use alcohol, convert it in their system, so they get exposed to it. All right. The mutational forms that have this intolerance for alcohol, they're fine, and unless they get into a system where there's lots of alcohol. If they get into a system where there's lots of alcohol and they lay their eggs, the eggs essentially won't exist, right? So it's conditional. It's conditional on whether or not they actually get exposed to a level of alcohol. In fact, the mutation, one of the common mutations, it only takes about 4% ethanol for it to be lethal. Okay, but if you have the mutation but you never get exposed to the either the low temperature or in this case the amount of ethanol, then it's not an issue for you. Okay, so it's conditional on whether you're even exposed. Some cancers that people have, we have the mutations that are highly correlated with certain expression of certain types of malignant growth. But if you don't get in the right environment, or the wrong environment might be the correct word, you don't necessarily express it. Okay, you may have it, it may have the mutational forms, but you don't necessarily express it. Okay, that's not all bad. There's, it's been actually used to deal with some problems. There's a whole um, sort of design system known as RID, release of insects with dominant lethals. And there have been all kinds of ways when we try to control insects like screwworms and other things. One of the things we do is we have done is sterilize a bunch of males, release those males into the wild. Once they breed, a lot of times insects only breed once. They breed with these sterile males, and you can reduce the population. But sometimes this doesn't work very well, and so there's been this whole system developed that is aimed at dengue fever, which is very common throughout the southern part. Actually, it does occur in Alabama, but it's found more in Mexico and northern parts of South America, as well as in Africa and other areas. And very problematic. Well, there's been a development of a sort of genetic engineering of some insects, in this case, mosquitoes that carry or vectors for it. And these are set up so that they have a dominant lethal with them. Now, it's a complicated system. We don't have to learn any of this. I'm just trying to show you how this can be used. But basically, what you do is they go in, they do a genetic modification by adding a certain type of promoter. Okay, the animals can be raised. The mosquitoes can be raised in the presence of tetracycline. So if they're in tetracycline, they don't have any problems. Okay, but if they don't, which they wouldn't in a natural environment, then they go through this process where they go through some specific splicing. We'll talk later about gene splicing. But what happens there is that it affects the females and not the males, right? It kills off the females. So the males continue to breed and all that. So what you end up with are these males that have this dominant lethal, okay? But if you raise them in tetracycline, they survive, they can function. When they mate with a normal female in the wild, then there's no offspring produced. When you take a female and a male or two that have these mutations in them, the progeny then can carry on that mutation into the next generation sometime, and then or they may die off, and then of course normal ones produce. So what they're doing is releasing this dominant lethal out there and kind of using it in a conditional way where if you have exposure or not exposure, you can produce these animals, release them, and once they get out there in nature where there's no tetracycline, then you end up causing production of no offspring. So you start to slowly reduce the population in these areas by having these sort of natural systems where you've introduced a non-natural variant, a dominant lethal into the form. Okay? Just an example that it's not all bad. All right, we've figured out how to use some dominant lethals. All right, so genes cheat are some silly way to say that. Delayed expression and conditionals are just a couple of examples. And what else can they do? Well, there's this whole idea of these sublethals and all that really falls under what's known as incomplete penetrance. And penetrance is a term that we haven't talked about yet, but it applies to all allelic systems, all right? Penetrance is the proportion of those genotypes that actually express what they should, okay? Meaning that if you're big A or you're big A little a, like, in anything, right? So let's go back to maybe snapdragons or something. Go back to incompletely dominant. Sometimes in those systems you have big A supposed to be red, or big R, make it red. And then big RW, right? So RW, which is supposed to be pink, but instead they're bright red. They look just like the others, but it's only a few of the individuals, right? So it's called penetrance. So not everybody always expresses that, and that's certainly true for lethals. Just because you get it doesn't necessarily mean you're going to express it. So we talk about the amount of penetrance, and we talk about, particularly in things like genetic screening, we talk about the level of penetrance that you might have. You screen someone and they find out that they have a marker for some genetic problem or some future problem, but you have to tell them, okay, it's penetrant, right? What's the penetrance of this? And some we know that the penetrance is really strong, and we pretend for these Mendelian problems that you've been doing, and basically all of the problems, word problems that you're working out of the book, there's 100% penetrance, right? So you don't have to worry about, well, every now and then I get a het that doesn't show up like it's supposed to. But in the real world, that's exactly what happens. Now, that can be because of environment, it can be because of all sorts of things. Very, very important in things like genetic screening, because these markers a lot of times aren't perfect markers for, and it can't tell you just because you have the marker doesn't necessarily mean you're going to get the disease or you're going to get the problem, right? And that comes into play if you are tested for maybe breast cancer and you see that you carry one of the allelic forms that is associated with breast cancer. Do you have a radical mastectomy when you're 20 years old or not? Well, it depends on how penetrant that genetic counselor feels these things are, and your physician. So there's, lots of decisions, but kind of have to ask, okay, what should I do, and how penetrant is this particular allelic form that you've identified? Okay. Well, that's dominant lethals. Again, they're not that common out there, but they do exist. I think it's important to talk about them. So now what we're going to do in the next part 2 is we'll come back and talk about recessive lethals, which are actually very, very common, and then talk a little bit about how some crosses are made and do some other things with those.